So we're going to start looking at how blood is used in a crime scene investigation, and there are two main ways that it can be used. First is going to be to extract DNA for analysis, or it can be used as physical evidence when blood spatter is studied. Whereas DNA or splatter pattern can give information about the truthfulness of an account by a witness or a suspect. It can also provide information about and even potentially. Our understanding of blood began in ancient times and still continues to grow today. So let's look at blood as a tissue that circulates around and throughout the body. It's composed of three major components. So shown above, respectively, are the red blood cells. And I'm sorry, I'm jumping ahead of myself. Sorry. The whole blood itself carries cells as well as plasma. And that's the fluid that has your hormones and your clotting factors and also transport all of the nutrients for you. Within that, though, the cells that exist include the red blood cells that are responsible for carrying oxygen to the body cells and carbon dioxide away, the white blood cells that fight disease and foreign invaders and alone contain cell nuclei. The white blood cells are the only ones that contain cell nuclei. That's very important. And then platelets. These aid in blood clotting and the repair of damaged blood vessels. Now, blood typing itself is a lot quicker and less expensive than DNA profiling. It produces class evidence, but can still link a suspect to a crime scene or exclude a suspect. For example, when we look at the constituents that compose the blood, if we were going to look at a single microliter of blood, the red blood cells make up 5 to 6 million components of that microliter. The white blood cells make up somewhere between 5 and 10,000, and the platelets are 250 to 400,000. So which cells do you think are involved in forensic analysis? Why do you think that? Hopefully you said the white blood cells and the red blood cells. So let's check this out. In 1900, Karl Landsteiner discovered that there was more than one type of blood when the blood of one person did not mix freely with another. Blood types, we found out, are determined by proteins that are embedded in the cell membrane on the surface of the red blood cells. So you've studied that there are marker proteins that identify every different species of organism, and you've studied that when you studied cell membranes in biology. Now, later, in 1901, Landsteiner identified what we call the A and B cell surface proteins. So what that tells us is on the surface of somebody that has type A blood, somebody that has type A blood only has A-type markers. Somebody that has B blood only has B-type markers. But the most rare type of blood is AB, and that's because those individuals have red blood cells that have both A and B markers on their surface. Type O blood doesn't have any surface proteins identifying which type it is. So it's almost like they're a zero. They don't get anything. And notice how that's 43% of the population in the United States. Now, you'll notice that there's something else written down there at the bottom. And I want you to note that the Rh factor is another type of protein that we're going to talk about in a minute. But 85% of the population in the United States is Rh positive. So A positive, B positive, AB positive, and O positive versus the negative. So let's talk a little bit about these A and B proteins. There are two proteins that are embedded in the cell membranes of the red blood cells and are called A and B. Somebody may have the A proteins, the B proteins, or both, or neither, and that makes them type O. The Rh factor is another blood protein, and people either have it, making them Rh positive, or they don't. That protein doesn't even exist on the cell surface, and that makes them Rh negative. So it's estimated that 85% of the human population has that Rh factor protein. Oh, excuse me. So when we name blood types, a lot of people refer to their blood type as A positive or AB negative, and that plus or minus is referring to that Rh factor. So, given the frequency of different genes within a population, it's possible for us to determine the probability that a particular blood type will appear in a particular population. If we think about somebody that might be AB negative, we already know that Rh negative is not very probable. AB is not very probable. That's probably going to help us indicate a particular person within a population. So to determine the probability of two separate events, it's necessary to multiply their individual probabilities. Now, you've studied this in math before, right? By identifying additional proteins in the blood as evidence in the sample, the investigators can actually limit the size of a suspect population and actually help identify a suspect. So the more events that they multiply together, the smaller the number is going to get, the more likely it's going to indicate a particular person. Now, there are three different types of blood typing. There's one for the A protein, one for the B protein, and then one for the Rh factor. So there's literally three separate tests. Now, when we look at the percentages of the population, we find that 42% of the population has the type A protein. B is 12%, AB is 3%, and O is 43%. Now, I've already said that Rh positive is 85%, so that means that Rh negative is 15%. Make sure that you have these written down in your packet. You're going to utilize these numbers quite a bit in our probability problems. Another blood typing test that we can do is for what are called the M and N proteins. Now remember, you inherit an allele from each parent. So it's almost like getting dominant versus recessive alleles. So you can inherit homozygous for M, getting both M's from both parents. You could get one of each, be heterozygous. You could get an M from one parent and an N from the other. Or you could inherit two N's and be homozygous for that N, Nancy allele. So we've got 30% for MM, 48% for MN, and 22% for NN. Now, there are additional enzymes and proteins that are included in identification. Some of them include phosphoglucomutase, or what is referred to as PGM, AK, which is adenylate kinase, SD is esterase D, and HP is haptoglobins. So those are just some that you're going to come across in our problems. But here's the thing, what we need to do is we need to go through and see if we can do a calculation. So if you were to look at the population, what percent of the population would have A positive blood type? So what I need to do is I need to convert the percentages to decimals. So I look at what's the probability of having type A blood? Well, if I look back at the chart, it tells me 42%. So that would become 0.42. The probability of being positive is 85%, so I convert that to 0.85. Again, I have to multiply the individual events to get the combined information. So I have to multiply those two decimals together, and then I can convert that back into a percentage by multiplying by 100. So about 36% of the population would be type A positive. So here's what I want you to do. I want you to take a moment, and I want you to see if you can do these two challenge questions, and then we'll go over them together. So for the first problem, I'm going to try and draw on here, so bear with me. For the first problem, the probability for getting type O is 43%. So I have to take 0.43, and the probability for getting type negative is 15%. So then I have to take 0.15, ooh, that looks bad. And then the probability of being MN is 48%. So I have to take all of that times 0.48, and I get an answer that is 0.03096. So am I done? Nope, still got to multiply by 100. So my answer, sorry, this is really bad, is 3.0, my zero didn't show up, 0, 9, percent. Can you tell that that's a percent sign? It's not showing up very good. There we go. So that's what my answer would be. I know it's hard to see, but now see if you can do the next problem. Okay, so this next problem is a little bit more difficult. So on this one, type A is 0.42. Being N blood protein is having NN, so that's going to be 0.48 times the HP minus 1 is 0.14%. Not percent, sorry, 0.14. The RH is 0.15 because that one was negative. And then the PGM2 is 6%, so then my final is 0.06. Alright, so when I multiply all of those together, you should get 0.00, oops, 0, I don't know why this is acting the way it is, and then 2, 5, it's not a 5, that kind of is. You'd think Mrs. Learned was still in first grade by the way her math skills look here. And my answer then, because I still have to multiply it by, good grief, by 100, why is my computer doing this to me right now, is 0.02, really, that's a 2, people, 5, 4%. Really doesn't like me. Now, when you think about the answers for those, which one would you want to have in a court of law? Okay? Because if you think about it, I would rather see the lower number, the lower percentage. That means it's less likely for someone to have committed the crime. So what does that tell us about the number of blood proteins that we're testing? In the first problem, we only looked at 1, 2, 3. In the second one, we looked at 1, 2, 3, 4, 5. So the more blood proteins that we study, the larger the number, the smaller the percentage that it could be somebody different. Now, on the back of your packet, what I want you to do, there's a blank page. Record these additional blood proteins and their probability because you will need them for the probability problems that we're going to work on in class. All right, see ya.